A very warm greetings to all the viewers. I am Dr. Dhruil Brahmbhatt, working as an assistant professor in Department of Microbiology in LG Institute of Applied Sciences situated in Ahmedabad. Today, we are going to discuss about direct repair mechanism. This topic is also included in Semester 5, Paper 301, Unit 3 in Gujarat University syllabus. Before we begin, it is important for us to know a couple of things. First, what is mutation? And second, how does a cell cope up with mutation and what repair mechanisms they adapt? Today, we are here to discuss one of many repair mechanisms that occur in microorganisms, namely direct repair mechanism. We all know a cell is a prolific mode of replication machinery and it functions efficiently and rapidly. However, due to rapid replication and synthesis of nucleotide sequences, a cell may sometime undergo alteration or error in its replication mechanism. Errors in replication can alter nucleotide sequences and generate variety of mutations. Nevertheless, a microorganism must be able to repair these alterations in the nucleotide sequences that might be lethal. The altered or damaged DNA is repaired by several mechanisms apart from proofreading or exonuclease activity of DNA polymerase III, which removes incorrectly incorporated nucleotides immediately after the synthesis of new chain. The damaged DNA is repaired by following repair mechanisms. First, excision repair, which includes nucleotide excision repair, base excision repair. Second, direct repair or photoreactivation. Third, mismatch repair. Fourth, recombinational repair. Depending on the type of damage, a cell adapts a specific repair mechanism. In this lecture, we are going to focus on direct repair mechanism, also known as light repair mechanism or photoreactivation. The main objectives of this tutorial will be to understand photoreactivation, formation of thymine dimers, enzymes involved in photoreactivation repair, and mechanism of photoreactivation repair. Photoreactivation is a type of DNA repair mechanism present in both prokaryotes and eukaryotes. It is recovery of UV irradiated damage of DNA by visible light. As the name suggests, it is a light-dependent process and hence also termed as light repair mechanism. In this method, the cell repairs DNA after it has been exposed to UV rays. The UV rays are lethal to cellular DNA since it induces structural lesions in the DNA with the formation of pyrimidine dimers. Photoreactivation process removes the pyrimidine dimers without altering other nucleotides in the DNA by using energy obtained from visible light. Light repair mechanism is the first discovered repair mechanism in the cell. In 2015, Nobel laureate Professor Aziz Sankar was credited for the discovery of photoreactivation mechanism in cell. As mentioned, exposure to DNA, as mentioned, exposure of DNA to UV irradiation causes pyrimidine dimer formation. Among different types of pyrimidine dimers such as cytosine-cytosine dimers, thymine-thymine dimers, and cytosine-thymine dimers, thymine-thymine dimers are the most common one. The two thymine residues adjacently situated to DNA strands forms covalent bond with each other, and this leads to structural lesion in the DNA. Thymine dimers are also referred to as cyclobutane photodimer, CPD. When the DNA is exposed to UV light, the double bonds present in thymine residues between C5 and C6 of both adjacent thymine breaks in order to satisfy the valency of carbon atom. The electrons are shared between C5 of one thymine and C5 of adjacent thymine. Also, C6 of one thymine and C6 of neighboring thymine. Due to such sharing, two covalent bonds are formed between two thymine residues which are referred to as thymine dimers or CPD. Because of the dimer formation, a structural anomaly is created and DNA lesion forms, which may lead to mutagenic effects. Now let's see a short history of photoreactivation. In 1940, scientist Albert Kellner reported that UV-induced damage can be recovered by treating the cells with visible light. He termed this phenomena as photoreactivation. Later, Renato Delbecco suggested that the photoreactivation may be an enzymatic process dependent on light. Stan Rupert further proved that an enzyme was responsible for the repair mechanism and named the enzyme as photolyase. Now let us understand the mechanism of photoreactivation. As previously discussed, it is a light-dependent process. The enzyme responsible for carrying photoreactivation is photolyase, a flavoprotein which contains two chromophores, which absorbs light energy from visible light. The chromophores act as catalytic cofactors. The two chromophores are fully reduced FAD and a folate group. The fully reduced FAD is essential for the catalytic property of the enzyme. The photolyase enzyme absorbs light at 300 to 500 nanometers and gets activated. The light activated enzyme splits the dimer by local free radical mechanism via transient electron transfer method. First, the photolyase enzyme recognizes the thymine dimer lesion in the DNA and binds to it. The enzyme then absorbs visible light, which results the activation of FAD. The excited FAD transfers electron to thymine dimers. This electron interacts with thymine dimer, which results into charge separated radical pair. The unstable radical pair suddenly splits and the covalent bond breaks. The electron returns back to the reduced FAD once the thymine dimer is repaired, and the photolyase leaves the DNA. Thus, photolyase repairs the lesion created by thymine dimer by utilizing visible light. Similarly, methyl and other alkyl groups that have been added to O6 position of guanine can also be removed with the help of light-dependent enzymes such as alkyl transferase or methylguanine methyltransferase. Thereby, damage to guanine from mutagens such as methyl nitrosoguanidine can be repaired directly. Now let's summarize this topic. Photoreactivation or light repair is a direct reversal method of DNA repair. Neither any templates are required nor any nucleotides are removed in this method. The pyrimidine dimer, which are formed as a result of mutation, is corrected via energy utilizing visible light. The light activated enzyme splits the dimer by electron transfer from FAD. Now let's look at certain interesting facts related to this topic. Direct repair is not a universal repair mechanism. Direct repair is completely absent in placental animals, including humans. Placental animals, including human beings, use nucleotide excision repair to remove thymine dimer. So here I conclude this session. I hope at the end of this video tutorial, we are clear about direct repair or photoreactivation. But a brief idea about formation of pyrimidine dimer, enzymes involved, and mechanism of photoreactivation repair. I hope you find this video useful in your studies in understanding the concept of direct repair. Thank you.